by Randall W. Nelson of the Biodesign Institute of Arizona State, and he will be discussing using high throughput mass spectrometric immunoassay in biomarker development. Randall?
All righty. Or another way to put it, it'll be the last talk. What do we got here? I'm just going to run Richard's slides backwards. Nah, I brought some for you. Hold on here. I got it. I've run one of these before. All righty. So, timer's on. Launch. Okay, so I feel like I'm under pressure. I'd like to thank you and Marvin and the rest of the organizers for having me here. This is quite an honor, and hopefully I can keep you awake for about half an hour here and tell you kind of the way I viewed MALDI. We've seen from Peter on out to Franz in particular, and then Marvin, and everybody who's sat here has seen that we've kind of evolved from before to the infancy of MALDI to maybe some future applications, in particular what Richard just showed you. And I think that's maybe where I fit in. And I got to say one thing too. You guys brought it up. That was, and I'm a little younger than you guys, but I will view that period of time as probably the funnest time in my entire career. It's a weird way to describe it, to watch the evolution of this big molecule, this lighting mass spec at fire anew. It just, I can't even explain it, but I hadn't even thought about it. It is really fun to have worked with you guys, at least a little that I have. So what do I do now? I just sit up here at this cabin, right, and I think most of the time. So to get away from my day job, which is a scientist, or at least I thought it was until I got deep into ASU and I'm more of an administrator now, it seems. I go up here and sit around and contemplate and build things. And fitting for this talk, I built a hot tub time machine. You guys ever seen this movie? Yeah. I'll tell you what, I'd jump in that thing any day, twice on Sunday. Takes you back to 1987. If you follow this movie, it's a big hot tub that you sit around and drink in, and it takes you back in time. And this one almost works. I've almost got there. And what I did is I went back to that star right there, which if this thing's still working, Richard, or did it break? There we go. Oh, it's red. So I kind of came into this right about the cusp of when MALDI and electrospray were coming on. So what I was doing for my thesis was something akin to MALDI, but not as good, quite frankly. But it was a neat project, and it got me in league with Marvin as a first job to start making these MALDI instruments, right? So you can kind of look at topical errors and what I'm going to call protein mass spectrometry. I think Peter did a very good job of explaining that we're trying to climb this bigger and bigger molecule mountain for the longest time. And then all of a sudden you can, and this was the fun part of it, right? Right in here, is that people were finding out what you could actually do with a protein in a mass spec. Whereas the day before you couldn't do it, now you can, and it just opened up this whole world of opportunity. But kind of disappointingly, it lost a lot of its topic, right? People don't look at big molecules a lot of times. They chew them up into protein or peptides and do proteomics, right? And kind of the irony here is that you've taken something you've waited pretty much for 100 years to do, and you've turned it kind of backwards. That's just me speaking, right? These are my ideas. And people now are doing proteomics, and we do see this all the time. And you guys want to know something that's kind of fascinating to me? If you PubMed proteomics or proteome, kind of put a star right in there, and look at the highest cited references in proteomics, they have nothing to do with what they found. It's how you do it. It's either a database search or a new digest method or something down the line. It has very little to do with the biology that supposedly you're studying. But nevertheless, we jump on over into this, if you're running out of things to do with proteomics, let's just call it a different term and call it biomarker discovery, right? And if you're in on this, don't go too long before you figure out that in order to have any discovery be meaningful, it has to be developed, right? And so there might be a technology difference here. And obviously leading to what you'd say in a hard word is diagnostics. This would be diagnosing people with disease, right? And so in a roundabout way, since that first day, at least when I was a star, I guess, I've been working on mass spec-based protein diagnostics, and it's growing in favor. This is actually thus the shading right here. And so we might want to talk about it and it kind of in isolation. And we've had some kind of, if you will, ricochets off, some tries at this. One was CELDI, and I bring this up because it really does use the capabilities of MALDI, right? In kind of the sorption SPE phase, this would be these targets are doing something for you. You're profiling one blood sample versus the next. It taught us a number of things. One is, the good things it taught us is that you could differential profile and maybe get some disease states or signatures on disease. Taught us to use pattern recognition and, if you will, multivariate index analysis to try to differentiate between diseases. Because of a couple of different faults in it, there's no identification of any of the proteins other than just molecular weight, and it was really restricted to high abundance proteins. It didn't really go as far. And I'll say offhand is that conceptually it was good. The instruments punched their weight. They did what they were supposed to do. But in actuality, the investigators didn't really know what they were doing with this, right? And I equate this, if I got a DUI, right, if I'm driving down the road drunk and if I got pulled over, instead of giving me a ticket, they give my car a ticket. So the whole community actually beat up on mass spectrometry for what I consider kind of unfair reasons. It's just a mass spec. It does what it's supposed to, right? Except they didn't do it quite as well. Nevertheless, it taught us a lot, right? Oh, I've left sequenome out here too. That's just nucleic acids. I think Franz is you were at one point in time very involved with those guys. But they've done a bang-up job on taking nucleic acid MALDI out into the diagnostic world. This, for those who aren't aware of it, last year, at the end of last year, two companies, Bruker and Biomarker, if I pronounce that inaccurately, forgive me, got FDA approval on essentially using the profiling approach to differentiate different microorganisms in your body. All right? And the way this goes is you grow them out, you throw them in a machine, you get a profile for one, you put that in the database, secure it in the database, and then add 2, 3, 4, 5 down the line. In my opinion, this is the biggest thing since MALDI to hit mass spectrometry, and here's why. Is first off, it's an FDA-cleared device for using MALDI, right? So Franz, on your ... it's never going to end. It's going to be there forever, all right? Another thing, too, that a lot of people won't view as important as I think it is that it goes at this in not a preloaded way. If you're familiar with in vitro diagnostics, they ask the question, the formal question, is this molecule here, and if so, how much, right? So instead of asking, what this thing does, instead of asking, is it this germ, that germ, another germ, so on and so forth, it asks the question, what is the germ, right? And if it's in a database of up to about 200 different species, it'll come back with an answer. And this is just a whole new paradigm in diagnostics. What is it, rather than is it this? And hopefully you can view the logic there. In short, it actually tells you one of 200 different circumstances that your body's in, 200 different germs. I wish we had 200 different treatments, to tell you the truth, right? And also is predicate for anything else you might dream of that kind of revolves around MALDIs and multivariate index analysis. So it lays literally the foundation for additive assays, right? Richard, I've got you in here. You just did a better job than I could at explaining your own work. But the thing I really think is nice is it's label-free histology. You brought this up throughout the talk. It's just open-ended. What is it? Instead of histology going, is it this? Right? Because the mass spec's good at doing that. And so this is kind of the core of what I'm going to talk about for the rest of the time. Back in '95, we introduced mass spec immunoassay, and we've taken it, even though I don't say it, we've taken it into CLIA laboratories, and we've moved this off commercially to try to take it farther and farther into a diagnostic mode. I mean, FDA is really the way to start thinking about it. But when I start talking about diagnostics, because I get questions all the time, and I have for 20 years, why use mass spec, right? Well, why use anything at all? But if you can answer that, then somebody else is going to ask you a question about this, and finally this, and it costs too much, and what have you. And so these are my unified thoughts on any kind of diagnostics, but in particular mass spectrometry. And the first thing we got to do is we got to understand what diagnostics is, and it's not an analytical technique, it's the business, if you will, of the information arm of healthcare. That's how it operates, right? We view it as a physician gives us, takes some blood, sends that blood out for a test, particularly maybe at LabCorp. The test comes back, and then he'll cut your spleen out or whatever he's going to do based on that information. The bigger picture is it's a business. Samples go into this business, information comes out, and it's a for-profit business. So you have to be cognizant of all the way along the lines when you're talking about diagnostics of the final, if you will, trump card or veto. If you can't make money off of it, it's not going to fly. Right? It doesn't matter how good it is. Right? But you got to answer that question. I'll return to that at the very end. Produce large amounts of data without going broke is what I'm saying. Now, I'm an analytical chemist by trade, and we brought up quantification, if you will, with MALDI. And I just want to point out, as that IUPAC says, that first and foremost, a qualitative analysis tells you what it is, and then secondly, a quantitative analysis is based on a qualitative analysis. So here's what this means, is that most analytical approaches are qualitative, and then, if you will, quantification is a method built into the qualitative analytical platform. You got to view it that way. I teach analytical chemistry, and it's the only way I can explain it. Most analytical qualitative approaches can be made quantitative, but all of them with a calibration curve, for instance. But most of them aren't automatically just throw something in and it gives you an answer how much is there, like a balance, right? It just doesn't work that way. You build methods into it. So if your approach isn't quantitative, maybe you ought to try a different method, right? That's just what I got to say about that. Because I've heard it for 20 years, and we've been quantifying proteins for 25. And so, but nevertheless, too, if you back out one more step, and I kind of like this is kind of your deductive reasoning, inductive reasoning. All analytical approaches will rely on this sort of one of these three sort of, if you will, philosophies, right? And inductive is what ELISAs are based on. And what it says here, there's a lot of words here, but essentially an inductive assay is that if you walked into a room and somebody was laying there dead and the butler said or the wife said the butler did it, you would just say the butler did it and take him off to jail. That's pretty much it. You don't question it. He may be wrong, but you don't question it. You don't search for the false positive, right? Here you do. The butler's out of town. That couldn't work, right? And here, this is basically the wife's caught on camera. You just saw her actually do the murder, right? Mass spec immunoassay actually is the combination of all these three. We take the best of the inductive, which would be an antibody, which might be used in ELISA, but we don't actually let it produce a false positive, right? So if the antibody doesn't detect the exact molecule we expect, we denote that as a false positive on the qualitative aspect. Here we would figure out, if you will, this would be prove the true positive. Between the two of these, we'd have no false positives, right? And the two are different, quite frankly. And here we're looking for something we didn't anticipate at all. And this might be a variant of a protein, right? Which, like Peter brought up on the post-translation modifications, are all over the place. Your antibody may not have actually anticipated these things or may capture them and not differentiate the two. We published this back in '95, and so this is, in my hands, it's over 20 years old. And essentially, it's antibodies grabbing whatever you'd like to see, pulling them out. Here's one where we're pulling out two different antibodies, two different peaks in here. One's with an internal standard you quantify because you drug the internal standard through the whole process. You generate a working curve, and off you go, right? Then you write your paper. And what we did is we kind of kicked back and we saw what we were doing, and we were actually hitting limits of detection that are not quite as good. We'll get there, but we're pretty encouraging in competition with an ELISA. Remember, I'm not competing with mass spec. If I'm competing at all, what I'm trying to do is make a mass spec-based ELISA here, all right? We describe why it'd be useful. You may not need a monoclonal antibody, and particularly in a sandwich assay, you may not need two, right? You certainly don't need to. Radioactive material, which back in '95 was important on radioimmunoassays, don't need those, right? Could be multiplex. You could detect in interfering, i.e. you could have non-specific binding as long as it's not at the same molecular weight. You can get by with it, right? You can do the assay in a fairly short amount of time. And I'll show you a couple of things here, why this is important. Now I'm going to move up closer to now rather than '95. One of the tests we made up a couple of years ago is for parathyroid hormone, which is an 84 amino acid protein that swims around in your body. And over about 40 years, people have actually found out that it can be truncated here and here, right? So there'll be fragments of it. And because of those fragments, and in assay design for ELISAs, is sometimes here's what comes out of it. Because it can fragment, a test may, in most people, do this sometimes, right? I think that's kind of funny coming from the personal point of view, because if they ran this test on me, would I be the one that it might do it sometimes, maybe on Thursday? So essentially, you might as well say these tests we use in the hospital have all kinds of just, if you will, errors built into them. We've become so used to them that we actually just include it in our language. I think this is funny. I couldn't write a paper that said, this may work sometimes, somewhere, right? But this test is used every day. Well, what's the issue here is that you go in and you start taking a look at what's really going on with parathyroid hormone, right? By using an antibody that grabs this epitope out here, drag all the fragments out at the same time, and look at them in a MALDI. And it's not just 3 of them. I think there's about 10 of them or so, different molecular variants that can confuse an existing assay. And we've actually bootstrapped that over to an electrospray version of it too, in case you want to do MRM. But what it turns out is that just in one analysis, just going in and starting to look at this in large populations, by the way, about 500 people, you start to annotate this thing. This thing not only clips here, it clips here, and you get the picture here. We start to fully annotate this thing and look at the distribution of it in different patients, right? So there's at least a number of different plasma variants. Some of them can actually interfere with this assay, and then some of them, too, are actually more informative on chronic kidney disorder, right? So between stage 4 CKD and end-stage renal failure, some of those other fragments are actually the molecules you should be looking at. Here's another example of it, and just not to belabor it, but to give you a taste for in my mind, what we've been having to address in conventional ELISAs. You go out and you go through congestive heart failure. One of the tests that you'll run for congestive heart failure is for what's called BNP. And the assay is actually dependent on where these antibodies touch and feel. This would be individual from one manufacturer to the next, right? But over the years, the medical community has actually coined the term BNP paradox, and it's really this says is that your body's natural defense for congestive heart failure is to produce BNP. Right? And so what happens is you use BNP as a diagnostic, and you see this huge level of it. And the bioactivity says you should be actually decongesting, if you will, coming out of congestive heart failure. And so the paradox here is that, and then to make matters even weirder, BNP is a drug, so you give a little bit more, right? So I got 100, I give 1, and all of a sudden I get better. And so the question here is like, well, you got so much of it in you. Why is adding just a little bit more make any difference at all, right? Well, the real truth of the matter is that when you take a look at mass spec immunoassay, the results we get here versus an ELISA, the ELISAs are giving you a read that's 100 times higher than is actual in your body, right? So it's not a paradox. I'll say that in a minute. It's not a paradox. It's just that your assays are detecting something different than you think they are, right? And so if you've got 100-fold, you're off by 10,000% right here. And I've got 1 nanogram per milliliter in my body actual, and add 1 nanogram back in. You've doubled the concentration, and then you see response. So you can see how drug delivery as well as diagnosis are tied together here, right? Oh, and one last thing, too. This goes towards sensitivity, and I'd love to actually make MALDI more sensitive, right? That's going to help out quite a bit. But right now, in the way that we're doing things, this is a chart that Lee Anderson created, and this is just known proteins in the human body and kind of a range of the concentrations they're known to be at. We're right down here at the bottom. These two assays have lower limits of detections than commercial ELISAs, the best ELISAs out there, all right? And you get all the molecular weight information out of them. So it's true information and good analytical chemistry, I'd like to think. So we're seeing a lot of modifications here in these proteins, right? And here's kind of a good example of it. This ApoA1 swims through your body and works on cholesterol efflux, so it's part of keeping you healthy, so to speak. It's in your HDL, so keep it healthy. And if you just monitor this on its own, a lot of times there's this kind of a quandary. If my HDL levels look okay, but I'm still getting the effects of high cholesterol, and what's going on, right? Well, the one thing that can happen with this molecule, here it is kind of in a healthy form. This would be the full length. This is minus an amino acid down here, and here's a little bit of sugar on it, is that it can change because of the disease state, right? So diabetes would push you into oxidative stress. Here you have a bunch of this going on, and part of this was in your slide, Richard. Oxidative stress, and all of a sudden you've gone from really predominantly one signal to about a dozen of them, all oxides. Well, these things start to lose function. So if I measure this entire bolus here, green versus red, they equal each other, but function, this is really maybe about a third the function of this one, right? And like I say, this is important because when I start looking through populations, here's 96 healthy individuals. I'm looking straight down here, and I can see my heat distribution. These are the major forms of the ApoA1, the triox, if you will, and then these are sugar-coated over here, so the whole distribution is repeating itself over here. Long story short, when we start looking at what these proteins really are in populations, you see what I consider kind of healthy proteins and sick proteins, easiest way for me to explain them, right? So you might be sick with diabetes and you have this sick version of diabetic protein. It could come from cardiovascular disease too. The important thing, and this adds on, so this goes back into reading Peter's works for years and years, right, where you characterize these PTMs, which you're pretty much the godfather of all that stuff. Hopefully this kind of adds to it because what I realize is stoichiometry, these things in the biological situation is really where we start to get the telltale information of the diseases, right? So you not just laid the groundwork, you inspired us all, I guess is what I'm saying here. So I've got all of these modifications. What I'm going to do is I'm going to set this up and really start looking at sick people now. And this is just the easiest way for me to represent it. I've got nodes of disease, and most of us will know that we don't want to be diabetic. At some point in time in our life, hopefully we've started as healthy, but transpired or transitioned into diabetic population. And then once you're here, a number of things can happen to you. You can go into these comorbidities, like have a heart attack here. And I want to kind of emphasize that we're looking simultaneously at, we want to not be confused that we would see confounders, juxtapose plasma with serum. So here's a challenge group towards the healthy plasma, all right? Then this is all plasma over here. This challenge is here, but once I get here, right, which is diabetic with a heart attack, I have to challenge it with heart attack without diabetes. So every time I add a comorbidity, I got to expand my cohort. This whole thing expands geometrically. Hopefully that makes sense. What we're doing is we're trying not to view these diseases in isolation. We're trying to view them in the free range mode, what really goes on, right? I'm going to run about a dozen assays, of which, because of all those variants, I'll pull out about 140 pieces of data, right? And then I'll use that data because I know what it is, oxides or glycation or signaling, and there's some other axes I can build. But I'll pair it up. If I see something that's created by oxides, I'll take it and add it to, if you will, the oxide pool. And then signaling, you get the point here. So each one of these points is made up of about, at this point in time, it's about 21 determinants because I've reduced it down. So there are 21 pieces of information that go towards creating each one of these points. And my intent here is to create a biosignature that pulls these groups apart so I can look and see whether you're moving from healthy to diabetic, not just you're diabetic or you're healthy, right? and and ditto on the heart attack over here. The long story short is that you start to build these pathology-driven models, equate your PTMs to them, and most of these things are down at the, call it less than 10% level, the modulations that you see, right? So if you're kind of down there in what you consider as bad signal and noise, add another one, increase the signal and noise of that moiety, you get the picture. It's the same thing you're doing on your signal averaging on a MALDI instrument. And here's how it's kind of, this actually, we've moved this into these huge cohorts over here now because it worked pretty well in cohorts up to about 500 people all across the board. These are ROCs, right? So I can separate these things out, in particular down here, big gray space right in between here, where it's not that you're kind of diabetic or kind of healthy or overlap, that there's complete open range here. And where's this valuable? It's in what you consider describing pre-type 2 diabetes, so you can administer early drugs, if you will, earlier in the timeline of the disease. Build faster markers into drug trials right here. And also there's a FDA guidance to industry comes out about 6 years ago. It says you need to be very conscious of the effects of your anti-diabetic therapies relative to cardiovascular disease. So build signatures in here and see whether you're increasing the potential of having a heart attack. So now we kind of return full whatever. You guys still with me? Hang on there. It's going to end pretty quick. Okay. Thanks for the vodka, by the way. This is great. That's what's in here. No, it's water. Watered down vodka. What's that? W.C. Fields. Did he have somebody put water in my water? Yeah, I don't know. Anyway, here we go. So how are you going to do this and not go broke is really the question. One thing I left out is that after I worked for Marvin, I went back to university, and I was at the university for 3 years, and I started my own company, right? And that's a daily quandary. How are we going to actually survive till tomorrow? Anybody started their own company in here besides me? I know Marvin has. There you go. It's kind of brutal sometimes, right? And so, but nevertheless, you chisel your way through, and all the technologies that we're talking about, that's what we developed on out. The company was sold to Thermo 2 years ago, and in the meantime, I'd come back to ASU to set up a research lab, right? So now I got to buy the stuff I actually gave to myself at the company, but hey, it's okay. But anyway, when you're running your company, it turns out when I went back to academics, I didn't realize that they have a different way of accounting for everything, i.e., if it costs $1 to buy something, that's how much the assay costs, right? Nobody in academics actually does their books the way you got to do in your company. What, don't you get paid? So essentially what you find out is that people focus very much on their unit cost, but ignore their fixed cost. And really when you're running a business, you're buying time, right? Time equals money. Because I'm away from, if I was working there right now, if I'm away from there right now, the power company doesn't give me a day off, right? And I still got to pay myself. So you got to factor in on any of these analysis for the final cost or final cost burden, the fixed cost in here. And like I say, this is a strange territory for academics, right? So how you do that, and so I've set up two different kind of workflows here. This is a serial workflow where a sample gets run, if you will, sample workup time, and here's a piece of data, and during that time, only that piece of time, are you generating something that's a commodity that you can sell, i.e. this is the data, the information. Parallel workflow, you can see per unit time over here, I'm generating a lot more data. You'd like the parallel, obviously. The first thing we're going to do, so, and we're scientists too, I don't need to guess at this, so I went in the lab and I said, okay, IGF-1, insulin-like growth factor 1, is a FDA marker, approved marker with the CPT code of $64, right? I.e. every time somebody sells a piece of data, somebody somewhere is writing a check for $64 coming on back. You may not get it all. So that's the most any assay can cost me, hopefully a lot less. Then I'm going, if you're going to be in a clinical laboratory, you're going to have to handle at least 1,000 samples a day, right? Even though I'm not a clinical laboratory, let's see whether you can run 1,000 samples in a day, not just 100 and figure it out. Let's just see whether it's practical to do this, and what do we need, right? So the gauntlet is over 1,000 samples, 24-hour period, we're going to run this MSIA. We'll do it 96 at a time all the way through. And in the first iteration of this, we're going to run Bruker instruments, very good instruments. They're circa 2006, but they still work like a champ, right? They're a little slow on the laser rep rate, but they're very good machines. And what we did is it took us not a whole day, it took us 9 hours, right? And from this, I can actually do my economics on this stuff, right? How much did it cost me to generate each piece of data? Well, it took a minute of time for each piece of data that comes out. A minute in my laboratory costs $1.24. That's what it costs to run my lab, right? Plus $5, that's a tip I'm using, cost me about $7 total, okay? Well, if I was to run this out on an electrospray where I'm going straight forward here, just one after the next with a 5-minute turnaround time, which is pretty fast in just LC, right? That 5 minutes costs me about $7, even if I don't do a tip, if I just somehow magically got the IGF-1 almost for free for $1, it's still going to be $9. And the time right here, my unit cost, quite frankly, the way to think about it is my unit cost equal about 4 minutes of time, right? So anytime an assay is over 4 minutes long, it's cheaper for me, or I make more money if I actually increase my unit cost or cut that fixed cost down. Hopefully you guys are following me here. So that's what we're doing on the Brukers. Then I got hooked up with Marvin, and we got one of these units right here, right? And it's got a lot of good points to it, and we're making a whole portfolio of assays to literally, for the reasons I just said, can we make a MALDI diagnostic platform? And the data are the same as the Bruker, which I kind of considered at the time the gold standard, right? Oh, I got a little movie for you, and here's what I like. It's kind of boring or exciting or geekish or whatever, but it's not very long. This thing's actually running, and here's what I like about it. You can't really hear it is a problem. You'll see it. So here's what this thing's doing. It's on this spot right here. When it moves from one to the next, you'll see it go. Zing, when it gets all done. You'll see it move on over. There, so it moved over the next spot. So what that thing just did is took 50,000 laser shots and summed them up into one mass spec, right? And then 10 seconds later, it moved to the next spot. And you can see it's going to chisel its way all the way through that plate, right? And so what happens is every 19 seconds, this thing cranks out a piece of data, right? So my unit costs are almost nothing at this, or fixed costs are almost nothing at this point in time, but my throughput just on this machine, even though I ran like 400 samples here, so it ran 450 samples, I think, in 2.5 hours. That's start to finish. Remember, these are coming out of the freezer and then ending up in data. It's not just that, hey, let's see how fast this machine will run. It's the whole workflow I'm talking about, which is pretty good. It shaves $1 off because of the time on the previous thing, right? But the thing is with those 50, and Marvin brought this up, this with those 50,000 laser shots rather than about 5,000 when I was taking before, the precision of the data capture increases by about a factor of 2. So my CVs on my working curves are less than 3%, and the whole plate deviation, this between 5 different plates run 5 times, I think, or some numerous amount of times. Or, no, it's 5 plates. It's 96 samples run 5 times, right? And bang, 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 bang from start to finish. I end up with less than 4% standard deviation of all my data. And just as a side note here, this is better, cheaper, and faster, better analytically than the FDA commercial ELISAs, right? So with that said, in 2 minutes left, I'll sum it all up. I think you can do this. I think there's a huge future for MALDI. I think there's a huge future for all electrospray in, if you will, protein-based diagnostics. I think we're seeing the beginning of it, right? It really began 20 years ago, but people are starting to embrace it for all these reasons. Like I say, when you're talking about diagnostics, it's just not an analytical prowess type thing. You got to prove you can make money on it too. I'm sorry to say it that way, but that's people just going to shut the next guy. In your meetings, you might go through the scientists, but sooner or later you'll hit a VP of finance and they'll go, no, we can't bring that in because it's too expensive, you see. So you got to answer all these questions. One of them that a lot of people don't think about, because it's really easy to think that everything else exists out there, is that I told you about a better part of 24 proteins here, and these all are protein names. If I was to analyze those and these are things that contribute to those biosignatures, right? So I target ApoA1, and you saw BNP, and you get the picture here. And so these are things we run universally to create our biosignatures, but if I had, first off, the assays for the variants don't exist, right? But if the assays for the proteins exist on a conventional platform, I got to go out and buy 4 different conventional platforms for this, right? Because there's not just one ELISA vendor that will make an assay for all 24 of these proteins, right? And of course, like I said, we'd lose all the data that we really build the biosignatures off of them. So, you know, I'm not trying to convince you anything, right? I'm just trying to tell you what we do, and it's largely based on MALDI, and it's analyzing people. As people ask me what I do, I analyze people, right? And in the meantime, I make sure that I stay away from this cat. He guards my cabin up there. He's pretty cool. So I'll take any questions you might have. There, it just ended. I had 1 second left.
Good.
Yeah, how's that for timing?
Any questions? Well, let me just say it's been a wonderful afternoon of accomplishment, excitement, and enthusiasm, and that ain't bad. So, thanks to all our faculty.